So thanks for coming out on this windy, rainy night for talking about the gut and the brain. This is a fascinating area, and we're going to hear lots about it tonight. So I'm a microbiologist. I've studied all my life microbes that cause infectious disease, especially diarrhea. So that's why I have this really perverse view of the world, and that's why this talk is restricted for fecal content. So what I plan to do tonight is, given this mixed audience, I know there's public here and there's scientists, you're getting a bit of both. So if I'm getting a little technical and your public hang in there, if we do public hang in there, scientists will do a bit of both. So it's kind of a blend that I want to do tonight. What I want to do is I want to plant this really bizarre concept that the microbes in your gut actually influence your brain. And, well, let's have that. So I'm going to be talking about what we call the microbiota. Microbiota is basically all the living microbes in and on you. You sometimes hear this word interchangeably with the microbiome. That refers to all the microgenes that are in and on you. And the numbers of these things are absolutely astounding. There's about as many microbes in and on you as there are human cells in you. So I joke with my students, you're as much microbe as you are human. And they code about 100 times more genes than Homo sapiens in you. So I love to tell my students, these microbes have a big effect. So just because you can't see them doesn't mean they're doing anything. And that picture there, that was me, got bored in the lab one night. So I brushed my teeth and I scraped out my cheek cells, put it on a slide and stained it. That's all to do, I'm bored. I mean, look at it, first place, it's beautiful, sexy cells, gorgeous nucleus, You see all those other funny things on it. That, of course, is the microbes. Microbes are in your mouth and they're all over you. And as we'll see in a second, down in the gut, there's lots and lots of these things. So where are these microbes in and on us? They're basically wherever your body is in contact with the outside world. So of course in the mouth, you have the respiratory cavity, there's some. Your skin is like moist areas like the armpits, much better than dry forearms, for example, but there's still microbes there. They're in the lungs a little bit, and your genital tracts, there's lots of them, especially in women. But basically as you go down from the mouth down to the lower bowel, they increase in number, and by far the most is way down at the bottom in basically your gut, the largest colon. So the numbers are phenomenal. 100 trillion probably microbes on you. And just to put that in perspective, here's my first gross analogy. If you take a feces about the size of your little fingers, a little chunk of poo like that, there's more microbes in there in a gram of feces than there are people in this entire world. So think of the genocide you create every time you go to Washington. So what I want to do today is talk about all these microbes and some of the evidence that we and others are finding these microbes actually can influence the brain. And we'll talk about various things both in early life and late life, degenerative diseases and neurodegenerative diseases, and how these microbes are influencing it. And this is an exploding area. We certainly don't have all the answers, but there's more and more smoking guns and hints and things like this that are coming out. And ironically, I do many things when I work in asthma, malnutrition, diarrhea. I can't get anyone to come to lab work on anything other than gut-brain axis now. That's what everyone wants to study because they think it's such a cool frontier. So, as you know, you have a brain. It talks to the gut, tells you when you go into the bathroom and things like this. But we also know there's stuff coming from the bottom up, from the gut up to the brain. And there's been a lot of interest in how it works. Bottom line is, we don't know yet. That's what a lot of us are working on. There's a bunch of different ways. Microbes can make neurotransmitters, for example. They make a lot of the precursors for neurotransmitters, for example. They can trigger inflammation, which then triggers these cytokines and then move up and then start to affect the brain. Or they somehow need to be able to directly interface with the telephone line that goes from the gut to the brain, the thing called the vagus nerve, the big nerve there that hooks the gut to the brain. And there's these sort of three general mechanisms, the themes that microbes seem to influence the brain. But as I say, the details are really, really sketchy right now. And really, this is just an early area of science, and we really don't know, as the quote says, we really don't know a lot about this. So the first example I'm going to give is malnutrition. So that's when you don't have enough nutrients when you're young. This is the thing we started, funded by the Bill Gates Foundation, to really start to look at early childhood malnutrition. And at the time, there was no relevant models in animals to study this. And it's pretty hard to study starving kids. So we set out to basically develop a model to look at this. We developed this model. This is a mouse model. And basically what we do is we feed them, the green is a control, and those things, those big circles you see, those are slices of the gut in the mouse. And so the control, the top one, that's what a normal mouse intestine looks like when you feed it normal food. Then we thought, well, let's feed it malnourished food that doesn't have many nutrients, and we saw an effect in the gut. It was not huge, but there was some effect. But then we thought, well, these kids that are malnourished in these developing countries, they're always exposed to feces and unpurified water and things like this. So, and a characteristic is the presence of these fecal microbes in the upper intestine. So let's gavage in microbes from the large bowel into these things, as well as malnourished diet. And we did that, we got a remarkable effect on the intestine. The thing that says EE there, that's something called environmental enteropathy. This is a major disease in malnourished kids, and it really has a major effect on the intestine. So bottom line is, we got a mouse model to study this. We did a bunch of things, and this is just a diagram to remind me to tell you that this basically represents all the things you see in malnourished children. So basically, if you give a malnourished diet and you give them microbes from the top down that are normally in the intestine, you basically see things like you increase the intestinal permeability. That's a very hallmark of this disease. You see the changes in the microbe, we call this dysbiosis, that's screwed up microbes. We see that. We definitely see growth stunting. We see inflammation, which is a major marker in the gut. And as well as you saw the villus blunting, that's the lining intestine, gets much stunted in it. So well parameters is a really nice model to study malnutrition. We're working on various ways of malnutrition. But one of the major questions is, well, could this be having an effect on the brain? And it's well known that malnourished children, their brains don't develop as well. They basically don't reach their normal, or hopefully their potential mental abilities normally. And it's well known in kids that brains don't develop. So we thought, okay, we've got a model. Can we actually study what's going on in the brain? So to do this, we basically started to study a particular cell in the brain. It's called the Microglia. And this is what they call it a macrophage. Macrophages are cells that go around your body eating microbes and chewing them and digesting them. There's a certain subset in the brain called Microglia cells. So they're basically on the hunt for potential infections in the brain. So they also remove neurons if they're dying, for example. But interestingly, they're involved in synaptic pruning. That's code word for basically reprogramming and recutting the synapses. Basically is how learning works and how memory works. So they're also involved in this. And so to do this, we actually took a mouse, and you can then put in a fluorescent protein into the brain in these cells, so they go green, and you can have a look at what's going on in there. So the other reason we thought we were onto something, this is a paper that was published a few years ago, and what they noticed a real difference in these microglia in mice that had microbes in them and mice that didn't. So we actually can make microbes mice that don't have microbes in them, we call them germ-free. They're actually microbe-free. You birth them sterilely by C-section, and you feed them autoclave food, and you keep them sterile. So these are called germ-free mice. And it was reported that these things called SPF, that's the normal microglia there, and GF, that's your germ-free. When you look at these microglia, they're really different from the mice that didn't have these things in them. They also noticed that they were treated with antibiotics, that's the EVX panel there, treated with antibiotics. The microglia are different than these things, and you can reconstitute things. So we thought, well, I wonder what's going on in our malnourished model to see, is there any effect in brain development? So Kalinda Bauer, a graduate student in our lab, collaborated with Victor's lab in New York, and we just had a look at this. So in the top is again those cross sections, the control, the malnourished, and the one that had those bacteria gavaged in, and below are the microglia cells. So control, this is what microglia normally look like. And microglia are really antisocial, they don't like to touch each other, they kind of keep each other separate. You see that they have these nice little appendages that come out, each their own little world there. You can see when you get malnourished and eat, they get really screwed up. And this was not normal, and we were quite interested as well as surprised to see this, that basically there's really differences in the brains of these animals. So you can do funky things like calibrate this, you can measure things like endpoints and branch points, and these are all the measurements of these microglia that basically say they're different, they're really screwed up. And then you can see the color picture at the top does justice to it. You see how very, very different when you change this. And of course the microbes change doing this. Now I said these things were like macrophages, so it's called phagocytosis. That's a word for basically engulfing particles. That actually causes the formation of what's called a phagocytic cup that's underneath. So that's basically the cup that goes under whatever you're engulfing. And we did that, we saw that in these malnourished and in the EE, we saw a real huge increase in these phagocytic cups. And then that raised the question, we're microbiologists, right? We didn't know what the heck we're doing. We asked our brain expert quite a bit, well, what do phagocytic cups do? What does this mean? And he said, well, basically they're involved in synaptic pruning. They're affecting the neurons and these things. Okay, well, synaptic pruning is associated with brain learning and things. Are they actually being affected in terms of their learning and behavior type things? So that took us down. So remember, we're microbiology lab, right? I mean, I never thought I'd study the brain. I think work on diarrhea and other good things like that. And so they said, well, you have to do something called open field test. What the hell is an open field test? Well, you go to Walmart, you buy yourself a great big container here, you put little tapes on it, you buy a GoPro, and you basically put a mouse in this thing, and you make the grad students stay up for weeks on end watching these mice and then do these traces. Now these are mice. Mice like to stay in corners. You do not want to go in the middle of anything if you're a mouse, because you could eat it, right? So normal behaviors hang around the edges. And what we found is that when we do all these traces, you can basically see as the malnourished eat, there's more mice, they go to the middle. And the behaviorists tell us this is weird behavior, this is abnormal behavior. We somehow screwed up the behavior of these things in these malnourished things. Okay, that's interesting. Then what else do you do? Well, you do something called the Morse water maze. You go to Walmart, you buy one of those little swimming pools. And you fill it up with water, put skim milk in it so the mice can't see where platforms are. And then you put these little platforms in these things, the mice figure out where they are, and then you move the platforms. You figure out how long it takes them to find it, or they go back to where they are. The other thing we notice is that sometimes the mice will just freeze. They just say, damn it, I can't find this thing, I'm just going to run, or whatever. They just freeze. And so interestingly, with what we saw, you can see, again, with the malnourished and the environmental apathy, we saw a profound difference in their ability to freeze and not freeze. So I'm not going to go through all the details. The bottom line is a whole bunch of these barrages of tests of learning and memory and things like that. Over here we have something called the novel object recognition test. We put these things into a cage and you figure out, are they curious about these things or not? And again, you can see that they basically, the malnourished had differences in these things. Talked about the water maze and the open field concept. So the bottom line is that basically by this malnutrition and the changes in the microbes in these things, they're actually affecting how the brain learns. So we continue to study this and try to figure it all out. But you know, enjoy the science, you never know what you're going to do. I was at this meeting and I talked to someone else from UBC, Janet Worker, and she works on early language development in children. She gets the 6-month-old, 1-year-old in the lab with these funky electrodes on them, and they like dangle balls in front of them things, and they record all these things. And basically they look at how they acquire language and things like this. And so we're saying, well, collect the poo, right? I mean, what are these experiments do? You heard the microbes, what are the microbes kind of thing? So they've been collecting the poo of all these kids and putting it in the freezers. And we are very happy to take this poo, and then we figure out what are the microbes in there. And we don't have results yet. We've got the poo collected, and we're starting to get through these things. We should know fairly soon. But so the idea is, it's a very bizarre experiment, that maybe we can look at the role of early life microbes in actual learning and language things. And because it's known, you know, germ-free mice don't have microbes, their brains do not develop normally at all. That's a common fact. So one night I was sitting to think about this, I was sort of half asleep, and just suddenly said, bolt uprights. Oh my God, what am I doing? Because in theory, language is associated with IQ, right? And I think, my God, we could find IQ bugs. And what are the ethics of this? And if we actually did find these things, you know, everyone would want to know their kid, and got really scared. Said, well, no, we'll do the experiment, see what happens. So we'll see. We also developed a collaboration with Person McGill. Let's do early music acquisition. Same kind of thing, but looking at kids looking at music. And of course, we're collecting their poo too. One of the fun things we can tell about this study is that we can tell who's got a dog in their house. Because that's obvious when you look at the microbes in the poo. That's an easy one, but don't know what brain yet, so we'll work on it. So what about other things in microbes and brain? Well, there's all these interesting studies out here. Stress, anxiety, and depression. You know, all these are major effects in our society. If you take a stressed, an anxious, or depressed mouse or rat, do a fecal transfer into normal mouse or rat, you actually transfer stress, anxiety, and depression just by moving the food from one animal to another. That's profound. In the UK, there was a massive study looking at all the people who used antibiotics. And what do antibiotics do? They kill microbes, right? They showed that basically that if you, there was a direct correlation with the courses of antibiotics and your levels of anxiety and depression. And it's directly correlated with antibiotics. Not antivirals, not antifungals, but antibacterials. I had a picture a while ago of a kid with autism. There's really interesting but premature early findings coming out of autism. That there's many reports of one-offs, the kid's normal, has to have an antibiotic infection, develops autism. It's often a year and a half. That's when autism can show anyway. But anyway, it comes along, and they're convinced that it's due to these changes in microbes. And there's some parents, we have to get a fecal transfer our kid, we have to move feces from a normal kid to this, see if we can. And there's actually been a couple of really nice pilot studies out now that if you do a fecal transfer from normal kids to these autistic kids, you actually decrease their autistic symptoms by maybe remedying the microbes. And it's a very contentious area, and we can talk afterwards about the implications of this, because autistic kids don't eat right, so of course the microbes are different, etc. But there's enough smoking guns here. There's papers showing that in mouse models, that microbes seem to play a key role in autism. There. I'm not going to talk much about much, but microbes also help affect circadian rhythm, which is basically, you know, when you know it's night, when you know it's day. And for any of you that travel, you know what jet lag feels like. It sucks, right? So this crazy Israeli sent his grad students, there's a great deal from grad students. He said, I will pay you a business class ticket if you fly half around the world, collect your feces, and fly back again. How many grad students I used to turn down an offer like this, right? 18 hours on a plane, you just got to poop in a jar, it's easy, right? Anyway, so we did these kind of things, and we showed that basically microbes affect jet lag. They affect circadian rhythm. And these microbes are talking to our body and our brain, and the hope is maybe you take a probiotic, something or other, that actually maybe could decrease jet lag if you have to do it, sort of thing. So let's finish up on the other end of life and neurodegenerative diseases. So Alzheimer's disease, we all know Alzheimer's. Basically, it's a developed country disease, and it's associated with basically developed countries. You don't find it very much in rural areas, and ironically, rural India, which is the cesspit of microbes, it's a microbiome man from the microbe point of view. There's lots of microbes there. They actually have really low rates of these things, where we are a nice, pristine, clean society. We all use hand wipes and hand sanitizers and sterilize everything. Basically, we see lots of Alzheimer's. So there's hints that microbes are involved in this neurodegenerative disease. There's a really neat study I found that basically, if you remember anything from this crazy talk, just brush your teeth 3 times a day. Brushing your teeth 3 times a day in this study basically dropped your Alzheimer's rates by 65%. Wow. I get it right. You say, what? What does brushing your teeth have to do with Alzheimer's? And flossing, not in the study, you can't answer that. I have no idea. Flossing, I don't care. So this actually makes sense, knowing you know, because a key thing to Alzheimer's and many other aging diseases is you get this chronic low-grade inflammation caused by your body that basically pisses your body's inflammation off. This causes tissue damage, which then causes, in this case, potentially Alzheimer's. So when you don't brush your teeth, you have poor gum health. The microbes seep through, they get into your body, they piss off your immune system, you become inflamed, we call this thing inflammation. And this chronic low-grade inflammation is associated with many diseases, obesity, diabetes, etc., and Alzheimer's disease. And in terms of experimentally, there's some really good studies that have recently come out in animals of Alzheimer's. Basically, germ-free animals don't have microbes, they don't get Alzheimer's in the Alzheimer's model. Or if you treat them with strong antibiotics, try and clear out the microbes, again, avoid this Alzheimer's. So, but that's an animal model, so proof is still found in people, but anyway, there's certainly smoking grounds in this area. And the next talk, we'll hear more about nutrition in the brain. I'll just put this plug in, because whenever I talk about, when I think about nutrition of food, I think it's the microbes that are chewing it, because they basically break down most of the food you eat, and everything you eat comes basically through this filter of microbes. So this thing called a mind diet, it's basically It's the Mediterranean diet, whole Mediterranean diet, olive oil, fish, glass of red wine, be cool, live healthy, type thing. And then something called the DASH diet, the cardiovascular diet. Bottom line, this diet is eating everything your mother told you should eat for you because it's good for you, and everything you don't want to eat because it's too good for you. And so it's really simple. It's basically fruits, nuts, legumes, potash, all those kind of things. Glass of red wine a day. It's get away from the red meats, the high-fat dairy products like butter and things, cheeses, fried food, simple carbs, white sugar, donuts and things like that. Those are not good for you. So why should I follow this? Well, there's now been 3 beautiful studies out showing that by following this diet, you can drop your Alzheimer's rates by over 53%. That's huge. All you gotta do is eat healthy. And there's some good stuff on here. And we're not perfect, right? I'm just not gonna quite, I eat like a steak once in a blue moon or whatever. There's a 35% decrease if you sort of follow this trend, but not perfectly. And like I said, that is really beautiful for this. So let's finish up with another neurodegenerative disease called Parkinson's. And Parkinson's, that's the disease you have the tremors in your stoop, right? That's got to be a brain disease. Everyone knows it is a brain disease. Well, in the last 5 years, it's basically, there's been a double take, and now, for much of what they go through, we actually think Parkinson's starts in the gut. And this, quote, neurodegenerative disease is actually caused by initially microbes in the gut. So the way this got kicked off is that in Denmark, if you had an ulcer, they don't do this anymore. They used to cut the vagus nerve that hooks the gut to the brain to decrease the pain. So basically, there's no nerve going there, so it's not the standard protocol. They were cutting this vagus nerve all the time. And what they did retrospectively went back is they showed that yes, indeed, if you cut the vagus nerve, you basically had way lower levels of Parkinson. So what is cutting basically the telephone line from the gut to the brain have to do with Parkinson? And no one believed this study. I mean, you're crazy, right? But then there's been 2 more studies that are showing exactly the same thing, that really this actually does influence it. And so that started people thinking, well, maybe there's something going on in the gut that precedes the actual Parkinson's we see later. And I think most of this is covered in this next one. So interestingly, Parkinson's people have very basic problems with their gut. The biggest risk for getting Parkinson's is constipation. So that's screwed up gut, right? A huge risk is eating red meat. That's a major confound of this. And ironically, coffee lovers in the audience rejoice. Coffee causes a 20% reduction. What does coffee do? Ask yourself that every morning when you sit in your throne, right? Increases motility, keeps you going. So bottom line is, and these Parkinson's patients have all these things. They also have something called IBS, irritable bowel syndrome, which is another major gut problem. Another weird thing about Parkinson's is that you lose your sense of smell. This is something that's puzzled everyone, but it's actually led us to actually start to swab out the nasopharyngeal microbes in Parkinson's patients. Say, are they different than normal? Because that's close to the brain and maybe it's affecting the senses of smell. The other thing is, this all happens about 20 years before you actually see Parkinson's itself. And we currently believe that basically 20 years before you get Parkinson's, you're constipated, you're eating lots of red meat, you're not drinking your coffee or whatever, and the protein that's involved in Parkinson's is also in your gut and in the brain, something called alpha-synuclein. And what happens is protein gets misfolded, and eating this red meat and constipating and inflammation in the gut, it's going to cause this misfolding of the protein, and then we think this protein gets misfolded, and then it gets misfolded, working up the vagus nerve, kind of going up the vagus nerve, and then it gets to a place in the brain called the substantia nigra, where it gets misfolded, and that's the area of the brain that actually is destroyed in your Parkinson's, because that's the one that makes the dopamine production. So basically, you then destroy this. By the time you get to the brain, it's done. It's irreversible. You can try injecting stem cells, and you can't do it. But the hope is, well, maybe we can identify microbes early in life, and hey, you shouldn't really have microbes like that. You need to change it, sort of thing. And so we've been doing a lot of studies with Parkinson's patient things, and we can actually look for some of these microbes, and we can say, with a poop sample, we can tell you with 85% confidence whether you have Parkinson's or not, by looking at the microbes in your gut. Nothing to do with the brain whatsoever. So you can see with this microbe in the future, we could actually think about, maybe can we give people undoubtedly poop transfers, with particular microbe transfers or diet changes or something push your microbes towards a non-Parkinson type phenotype, and then so you actually get Parkinson later on. So it's early days, but it's really exciting days because in germ-free animals, here's a really interesting story. In germ-free animals, basically in models of Parkinson's, you can take feces from people with Parkinson's and their spouse that doesn't have Parkinson's, put their poop into those animals. If you inject the Parkinson feces in animals, they get Parkinson's. If you inject their spousal feces into them, they don't get Parkinson's in animal model. So very compelling evidence, at least in animal models, of microbes and what's in the gut is actually doing these things. So I can stop there. This is where I spent the last 4 years of my life writing books about microbiomes, how they influence you. The first one is called Let's Eat Dirt. For any of you planning on having a kid or have a little kid, read it. It's about how you can raise your kids with their microbes. We've sanitized the world too much. I think that's really affecting child's health. And then the second one I actually wrote with my daughter, who did a PhD in gerontology, and that's for the rest of us already had our kids. It's basically how to age healthy with microbes, and I learned all sorts of amazing things about how these microbes really influence the body as we age. And so I need to give a thanks to the people in my lab that did all the experiments here, and then people that we collaborated with. So thank you very much.